the slide set on the tissue culture. Let me pull that up. I was saying how tissue culture can help with if you have disease sweeping through the plant. Sometimes if you turn to tissue culture, that can help solve the problem. So this is the disease that I had heard about from a couple of previous students that are now working in the cannabis industry, and it is initially attacks the hop plant, Cumulus lupulus plant, and hop and cannabis are closely related. They're in the same family of plants, and so now it's jumped into cannabis. And so I've included some quotes here from some of the websites. Whoops. So basically, they're the biomass, overall biomass plant gets hit. The secondary metabolite products get hit, and so even the trichomes look malformed and not full of secondary metabolites. And so 50% reduction in cannabinoid and terpene production, that's pretty significant. The only way they're finding to try to permanently get rid of it is to use meristem tissue culture techniques. So they go into the apical meristem, the primary meristematic tissue that they've been using, because as I mentioned, I think last time, that viruses have a tough time for some reason getting into the meristematic region, especially the shoot apical meristem. They seem to not be able to conquer those cells. And so if you can get those cells selected and grown in tissue culture methods, then the risk of having viruses, or actually it's a viroid present, is greatly reduced if you use meristematic. And so, by the way, so it's different than a virus. A viroid is, all it is a small piece of RNA, but it's infectious. It's kind of bizarre. It's infectious, and it can infect plants. It does not infect humans. There's been no documented case of any viroid attacking humans. So specific to plants, and it's just a piece of RNA. It's kind of bizarre to think about this as capable of causing so much damage to the plant. So here you're seeing some differences in appearance, in coloration, chlorophyll content of the plant, and what they're finding is that it's everywhere. This viroid is everywhere, and it's jumping from hops to cannabis. And some people are referring to it as the COVID of cannabis. It's that serious and widespread. So really, really bizarre. It's just a single stranded piece of RNA. It has no kind of, a virus has either DNA or RNA, and then it's surrounded by some kind of protein coat. And so viruses are different. This is just, we say naked. It's a naked piece of RNA. There's no protein coat surrounding it. It's just a stretch of RNA. That's all it is. So it is tiny, tiny, tiny. Here's a, if you can even see it. I don't know if you can see it. It will virus not there in comparison, size comparison relative to a virus, a mold spore, bacteria, and then on the left is only part of a plant cell that they're showing over here. So this is the plant cell. So you can see how tiny, 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 and yet it can cause such devastating disease. $4 billion in loss, some are saying. So I wanted to add that in because I thought this was a really timely example of what tissue culture is capable of trying to fix. If we can turn to tissue culture and create plants that do not have this viroid. Now those plants, when they grow up, that doesn't mean they're not susceptible to it. That doesn't mean they're resistant. It just means they're viroid free initially. And I thought this quote was interesting. Hydroponic systems. There's a real risk with hydroponic systems because you don't have to have the roots in contact with each other to spread it. It can just go through the water, through the system. So obviously, if you are using hydroponic systems, it's a big deal. The lower picture is trichome. The left are healthy trichome, glandular trichomes, and then you can see how deformed and flattened they are, so they're not carrying the same amount of secondary metabolites. Okay, so I just inserted a few slides looking at this particular relevant disease currently. And then the other slide set is what we're going to continue talking on. I updated this one too on propagation and grafting. I added a little bit more into it and over the weekend. So that's what we're going to get into next. And so we've been talking about propagation methods and you know, stem propagation, stem cutting is probably the most common, but grafting is gaining in popularity, and it used to be more limited to woody trees and shrubs. That was kind of the go-to way of growing plants, the way you want them to grow and the way you want them to produce certain whatever yield you're trying to get. So apple trees are the classic example. Apple trees are, you know, if you've ever bitten into a delicious apple, if you take that seed and plant it, it'll grow into a tree and produce apples, but the apples won't taste anything like the initial apple. They don't breed true. That's a fancy way of saying it. They don't breed true. And so all those apples that you see in the stores are generated through grafting so that you can get that consistent apple. You won't be surprised by the apple if you do it through seed germination and growth. So there are several methods, and these three are probably the most famous and longest used: whip or tongue grafting, cleft grafting, and T-bud grafting. And so the whip or tongue is what you're seeing in that image, where you have these cuts being made, and then you have the scion is the upper part. So essentially, you have a plant really like this apple. You can take the upper portion of that plant, and that's called the scion, and then you make cuts to match up the cuts to the base of a rootstock. And so you take maybe a root system from another plant. And so you can have another variety of apple tree, for example, and put those two together. And if you get the fit well made by your cuts, you have to do pretty good, precise cuts. And then you can slip them together like that. And then you have to have a way to secure them like that. The whipper tongue is pretty decent because of the way you make the cuts. The cuts kind of press against each other. There's some pressure there between the scion and the rootstock, and that helps. But then you usually have to wrap it and to keep it held together. And you usually cover it then with some kind of protective wax. Not always, but that's, especially with trees and shrubs, you tend to do that. And the key thing here, really, if you don't get this right, it's not going to work. You have to get the vascular tissue of both the scion and the rootstock. They have to match well enough when you make the pairing like that and add them together and piece them together like that. They have to come in good contact with each other, because if you can't get the vascular connections melding with each other, it's not going to work. You're going to have both systems will fail, the scion and the rootstock. And so the vascular tissue matching and getting that to fuse is huge. So right now, there's a big craze. Have you heard of these tomatato, something like that? Is that what they're calling it? So on the left, you see a tomato scion, the top part, fused with potato rootstock. And so you put them together. And on one plant, above ground, you get tomatoes. Below ground, you get potatoes. I think that's pretty awesome. I love both potatoes and tomatoes. So I had an undergrad student do this in the greenhouse. He had a couple of plants that he successfully grafted and was able to get both the tomato and the potato produced by the end of the several months of growing. So it can work. This image drawing is just trying to emphasize, again, this idea of getting the vascular connection. And so this is taken from a published study that was done in 2015. And they're trying to show that when you first marry the two parts together, you initially get just, sealing up of the wound, just like when you get a cut on your skin, you seal over, right? You get some growth, you seal over. But then you can see internally what's going on here. They're trying to, the growth of the vascular tissue starts to connect. And so in this particular study, they found, was it 3 days? You get the phloem, actually phloem forms first. That's hard to say. Phloem forms first, and that connection. And then they found the xylem not too long after made a full fusion. And so they're trying to figure this out and track it step by step by step. How does this all work? But it's really quite remarkable that the plant is capable of fusing like that and then connecting its vascular tissue. And they're finding that hormones are involved in order to get this to happen, which makes sense when you think about hormones in general. This is not exactly to do with grafting. This is more of a wound response by the plant. So they made a wound, all this red area, that's a cut that they cut into the stem. And I just put this image in here to get you thinking about that fusion of the vascular tissue. So this rerouting of the vascular tissue around the wound. Again, and they are finding that whether it's grafting or a wound response, they find that this hormone auxin, so there's a group of hormones that are called auxins, and they are important in getting the vascular tissue to form and connect. And so you've already learned about auxins. For example, the stem cuttings that you take in order to get the roots to start to grow, the adventitious roots. It's often you dip the stem cuttings into a powder that contains auxins to stimulate adventitious roots. So auxins are good at stimulating growth. And in this case, it's used to reconnect to the vascular tissue. So here are common strategies. So the whip or tongue is the top picture. You can see they make some precise cuts, and then you kind of slip the scion into the cut area here. And so the two halves of the rootstock kind of press against either side of the scion, and then you wrap it to further keep that in place. The cleft, you just put a cleft cut into the stalk, and then you slip the scion in and wrap it. And then the T-bud, you actually cut a T shape into the rootstock, and this is done with, they call it T-budding because you just use a bud from the stem, and like an axillary bud region, and you slip it into the T-cut that you make on the rootstock. So these are traditionally kind of the most popular long-term, like historically moving forward. These are the ones that have been kind of the tried and true techniques. But there's another one coming on the scene that's more straightforward than any of these, and it's called the splice grafting technique. It's simpler because you just make an angled cut on the scion in the rootstock, and then you put them together and wrap them or clamp them. And so it's more straightforward than what I just showed you on the other slide. But it seems to be more commonly used currently across different plant species. And because it's so simple and quick, they're finding a lot of success in the family Solanaceae, which is tomatoes and potatoes and so forth. Okay, so those are big crops. Whoops. And so doing this splice grafting is quicker. and it just saves time, saves money on labor costs, and has a good success rate with some herbaceous plants in particular. And so I'll come back to this idea in a second, but no matter what method you're using, it's like, okay, well, why would you choose to graft? What are some of the advantages? And sometimes you have plants that take forever to root. So if you can just take the stem of that plant and put it onto a really healthy rootstock of a related plant, that's a good thing. They've been doing this for drought tolerance. If you have a plant that has a root system that's just well adapted to drought conditions, then using that as the rootstock makes a whole lot of sense. Disease resistance, they're finding if you have a good rootstock that has really good resistance to soil-borne pathogens, and then putting the stem tissue, the scion, on that from a plant that's less able to resist disease, that's a good solution. They use it for dwarfing in order to create dwarfing varieties of different species of plants. We've also seen that you can increase the biomass of what's produced by the scion if you do the grafting. So certain plants, they can increase biomass of like fruit yield, for example, like tomatoes, if you do some grafting. Improving the fruit quality as well as the biomass. And some studies have shown better secondary metabolite profiles with grafting techniques. And so the cannabis industry and other medicinal plants, they're looking at grafting more intensely lately compared to previous. They're just learning so much more about grafting. It's becoming more mainstream. And this last one, they actually use it to check for diseases. So this is more like if you're trying to diagnose whether or not you have a disease running through that you can't easily see the symptoms of in a certain plant. But so, for example, if a scion is coming from a plant that doesn't exhibit disease visually, what you can do is do this grafting technique, and it allows, if you do it right, it's a way to test for, is this plant harboring some pathogen that I don't really see the symptoms outwardly, especially with the naked eye. But if you do this grafting just right, it's called a culture index for disease rating. And so you can use it to assess whether or not there's some disease or pathogens lurking in there that you just didn't know were there. And so that's pretty laborious, and you have to really, usually you send this out for a business to do it for you, but it is something that's commonly done. So grafting is, there's definitely a science and an art to it, because you have to make the cuts just right and match everything up just right. And then you kind of cross your fingers that the vascular tissue is going to fuse. But it's, like I said, the use of it is spreading out into a lot of different crops and plants. It used to be more just for like fruit, apple trees and so forth, but now they're starting to get into it across not just woody plants, but herbaceous plants. So here on the bottom picture, you're seeing tomatoes. On the left were grafted, and on the right, these are heirloom tomatoes. And so the same species, but the plant on the right was grown from seed, and the plant on the left was through grafting. And you can see a big difference in yield of the fruit in this case. So it can also be space saving. You're getting per acre of field, you're getting way more tomatoes in this situation with the grafting technique. And so again, it can up your profit margin for sure. And so the cannabis industry and other industries that are getting into medicinal plants, they're really starting to take this more seriously and looking at what can grafting provide for them in terms of the product, whether it's a certain key secondary metabolite, or whether it be fruit or flowers. And so it is even being used to create just visually stunning plants. This is an image of succulents that have been grafted onto one big, like mother plant, so to speak. And so you can see that there's many, many different species that were grafted onto one plant. And so you can imagine you could ask a pretty high price for that kind of house plant to have. It's so spectacular looking. So there's those types of interests too, looking at landscaping and house plants, unique things that catch people's eye. So it's really spreading out. Grafting is becoming big business right now. And with herbaceous plants, the grafting, like I said, traditionally, this is actually thousands of years old. People have been grafting plants for over 4,000 years. But it's usually the woody plants that they would do this with. Now with the herbaceous plants, you can get hydroponic tomatoes. They do grafting on those. A lot of the tomatoes that are sold in grocery stores are grown hydroponically or in greenhouse systems. So this isn't hard to do in the controlled environment and systems. And the grafting has also really helped with soil-borne pathogens. If you can find a nice rootstock of one of the cultivars of plants, a certain individual of that species, they are able to resist disease and then use that as your rootstock. And that has really helped with not having as much plant diseases. And so with herbaceous plants, modified whip or tongue or the cleft is often used for that. However, there's issues in the sense that you can't get too wild. Like you can't get wildly different species to graft together. And that's mainly because they won't link up their vascular systems. It just won't work. And so you have to be a little careful about that. You also have to be careful about the size of the rootstock stem, essentially, and then scion stem. They can't be wildly different in size. They have to be pretty similar. And so there are definitely some things you can't do with grafting. And so sometimes you'll see, I've noticed this too on like certain landscape plants where the graft, where they joined up, you can see a swelling on a tree trunk, for example. And it's not, it's not a gall or a tumor. It's because they grafted and the graft wasn't perfect and you get this swelling. And that often causes a weakness in that area so that if there's like a windstorm or something like that, the tree would easily break off and fall over. So there are some things that you have to be careful about. Have any of you that are in like the medicinal plants or controlled environment, have you heard anything about grafting for cannabis? I've heard fair bit about tissue culture, but I've been seeing some more readings about grafting. And so I think that's on the horizon. It could go mainstream very shortly here. And in fact, let me show you a quick video. They're starting to automate it, or at least partially automate it. So let me show you this. Don't really need the volume up, I guess. No, that's not what I wanted. I wanted you to replay. Replay. There we go. So the plant on the left is the rootstock. The plant on the right is the scion. mean, who thinks of these things? To design that instrument, that machine, it's pretty amazing. I think they're both tomatoes. I think they're both, but they didn't explain in any detail. So that's they refer to that as partial automation, where you still have to have people standing there, feeding the plants in the machine. But I could see where eventually that could become totally automated. But yeah, pretty cool little invention. So and pretty precise, if you can get it all troubleshooted out, over and over and over, you have that precision of all the same cuts. And so anyway, something that I think will show up as a more mainstream thing in the near future. All right, so where were we? Okay, so that was really the last part I wanted to show you. And then often with propagation, I wanted you to also just appreciate the term micropropagation. They'll use that a lot. And whoops, that is, I'm going to stop sharing these share because it's ignoring me. Again, anytime I go out of PowerPoint and try to come back in, it doesn't like it. Okay. So micropropagation, just remember that that term is really talking about tissue culture, which is a form of propagation. It's just micro referring to the fact that you can just take a tiny bit of tissue or cells, collection of cells, and work with those. Okay, so that's it for the propagation grafting slide set. And so you've done some propagation in lab, and then the biotechnology slide set with the idea of the genetic transformation. Hopefully you'll take a swing at that tomorrow in lab. And then the tissue culture, we're getting that set up for you to do probably either next week or the week after. And so you'll get to take a stab at tissue culture as well. But I wanted to get into the next topic. So what we've been covering is looking at the different types of plants and a lot of plant anatomy. What is the plant body composed of? What are the different groups of plants? Now I'm going to get more shifted into how do the plants function physiologically, their basics of their physiology, and then how do they respond to their environment in terms of their physiology. And so that can have a huge impact when you're trying to work with plants and grow plants for a profit, depending on what you're trying to grow and what your profit is. You need to understand the physiology of the plants and how to manipulate that and what kind of environmental parameters can manipulate that. So I'm going to start with the basics of, we have to think about water and how the plants need access to water. Primarily, we're thinking about now the typical plant that puts its roots into the soil. So we're going to be thinking about soil growth of plants, not hydroponic. And when you look at how plants have to deal with acquiring water, they don't have the luxury of walking to the sink to get some water. They have to deal with, where they are rooted in place. How do they get access to that water? And they have a lot of forces that can either work for them or against them in this process, in terms of gravity and some other properties that can make getting access to water. Oh, I don't know. Is it? Well, let's get that in there. So we're going to dip down into the water, or I mean into the soil, and think about water in the soil. That's the first thing we're going to do. Let me. And then we'll continue on. After we cover that, we'll get into how does it act once it's in the plant. Okay, now is it there? If you refresh, hopefully it's there. Water in the soil and plants. Okay. All right, good. So, okay, water is amazing. It's really an amazing molecule. And so here we have H2O, and you have that partial positive charge, partial negative charge, and that has a lot to do with how it behaves. And with respect to other water molecules, so the negative charges are attracted to the positive hydrogen of a nearby water molecule, and so you have those attractions between the water molecules to think about, but you also have attraction between water and other things around it, like soil. And so really an amazing molecule. And actually, it's kind of weird, but we thought we knew everything there was to know about water, but some scientists are saying, oh no, there's a lot we don't know about water. And there's been a lot of really intriguing studies done, which I don't have time to get into, but if you ever want to read more about water in your off time over the summer, it's pretty kind of freaky how we really don't know as much as we thought we do. All right, but some things we do know. Cohesion forces are important. So what are cohesion forces? This is the attraction between water molecules. Okay, so they like to cohere together, water to water. You also have cohesion allowing for this surface tension force to develop. Surface tension, surface. So we're think about like a pond where you have a body of water and then the surface water molecules and then the air above it, right? So think about the surface water molecules. What we see happening is the very surface water molecules on that pond, for example, those water molecules are more attracted to the water molecules beneath them than they are to the atmosphere molecules above them. All right, so something funky goes on right at the surface. You have those surface molecules, water molecules, more attracted to the water molecules beneath them. That causes the surface to tend to contract and act like an elastic membrane over the body of water. And that's what allows those water insects to walk on water. If you've been to an inland pond and got down to eye level and watched those insects kind of skating across the pond, and yeah, like, why don't they sink? Why don't their little legs just go right through and sink? And it's because of the surface tension. Those surface water molecules are kind of contracting downward and causing this elastic membrane over the surface. So we're going to talk about surface tension a fair bit as well. And then tensile strength is another thing to think about, more so when we start thinking about water movement inside the plant. Tensile strength is going to come into play pretty profoundly. And so the definition of tensile strength is the maximum tension that an uninterrupted column of water can handle without breaking. Now, tensile strength, you can measure the tensile strength of anything, water, steel, whatever. And so, but the definition, if you're working with water, that's the definition. So the maximum tension that an uninterrupted column of water. What I mean by that, uninterrupted column, there can't be any air bubbles in the way. No interruption by air, no air bubbles, okay? All right, so there's going to be some terminology here. I think that most of you have had some of this terminology at some point in your coursework. That's whether in high school or freshman, sophomore level here. Another term to think about or force is adhesion. So when we think about especially how water is behaving in the soil system, adhesion is the attraction of water to the solid phase around it, such as soil particles. Or perhaps when you start thinking about when it gets into the plant body, then you think about water molecules being attracted to the xylem, the cells of the xylem. So remember, the xylem are the tracheas and the vessels in the angiosperms, and you have water inside those being delivered through the plant body, through that vascular tissue, through the xylem. So this lower left image is soil particles, and you can see the water, the blue water, is kind of clinging to the soil particles. So adhesion in that case, it's the water being really attracted to the solid soil. Now, those of you who have taken soil class or are in it currently, you certainly know that soil isn't soil standard. There's all kinds of different soils out there. And so this is just a glimpse of a comparison between a couple of different versions of soil mixtures. So if you have sand or fine sand, coarse or fine sand, silt or clay, you're seeing the particle diameter of those different types of soils vary tremendously. And that has a strong impact on the surface area as well. And so you have, depending on the mixture of the different soil particles, we're going to find that just because there's some water in the soil doesn't mean that it's always all of it accessible to the plant, because you have some forces acting on that water that the plant may not be able to overcome and allow for the soil to come into the plant. In fact, there can actually be conditions where, unfortunately, it's the exact opposite. You can actually have conditions where water is coming out of the plant because of what's happening in the soil. So it can be tricky for the plant, for sure. And so we're going to find, and I'm certainly not going to start going into teaching all about soils. That's for the soils class to do for you. But just be aware, of course, that soil composition is a huge impact on the availability of water and how easily it can come into the plant body or not. Right? So we'll have to keep that in mind. So what we're thinking about is a typical plant has its roots in the soil. The soil water is where, that's the version of water that the plant needs access to. The plant has to take that water up into its roots, get it into the xylem, and then that will allow it to course through the plant body. And then eventually it's going to exit out of the plant in the form of water vapor out of the stomata, the pores. All right, so it's a continuum from soil, roots, vascular tissue, out through the stomata of the leaves. All right, so soils, thinking about the pore spaces of the soil. The size certainly is super important, and the number of pores is super important for how much water will be there, how much is available. So clay soils are very fine particle size, like what we saw in the previous table. So they have tiny pores in between those tiny particles. Sandy soil, the soil particles are much bigger. The pores are much bigger. Okay, that has a big impact on water availability and how much water is being held in those soils. So clay can hold more water than sandy. And I think if you just think about this for a minute, you've probably had experience with this at some point, even with potted plants. Different mixture of potting soil in the pot can allow for that water to be retained or to flow right through into the saucer below. So we talk about this water holding capacity of the different soil types, and different soil types have different water holding capacity. So what you're looking at in that figure is a comparison between if you have a sandy soil or clay soil in like a narrow tube, and then you start to add water to the top of the tube, and then you wait to see how long does it take before the water comes out the other end. I think if you think about it for a minute, you come to the same conclusion that, yeah, sandy soil would allow for the water to flow through much more quickly. All right, so you have a different water holding capacity between that sandy soil and clay soil. Clay can hold more. but that doesn't always mean it's going to be super accessible by the roots. So when we look at what's going on in the soil and how it's moving toward the plant roots, we need to think about gravity, cohesion, and adhesion forces. So as you know, when you water the field of your plants or you water your potted plant, gravity is going to take that water straight down. But we also have to think about cohesion and adhesion forces. That's what helps the water go more lateral too, especially if you think about like a big arid agricultural field where you have definitive spots of the field are getting watered. Obviously, not just those spots get access to water. You have to have that water flowing laterally to some extent out into that field so that all the plants in that field can get access. Capillary action will come into play when you think about evaporation of water from the soil system, for example. And so capillary action is actually caused by both cohesion and adhesion forces. So that's what you're seeing in that right image with 3 different sizes of capillary tubing. And then you put them into a beaker of water. And you may have done this in basic chemistry class where the narrower the diameter, the adhesion and cohesion forces with that smaller diameter allow the water molecules to go further up the capillary tube if it's in a smaller diameter like that, so the far right one, compared to if you have a wider diameter, there's still cohesion and adhesion forces, but they don't exert as strong of an impact, and so you don't get as much of a lift or capillary action. And so you have to think about that with regard to soil pores. We also think about capillary action a little bit with the xylem too. Think about that. So capillary action really does help water move out from the spot of irrigation to get it to move in any direction. And it also allows for evaporation too. So that capillary rise moving straight up out of the soil, that's what we think about with evaporation going on. So lots of different forces to think about when we look at soil. And, you know, if you think about growing plants in the soil, the irrigation system that you employ has an impact on how well that water is going to move through the soil. And so when you start thinking about water movement in the soil, or in the plant, the way we think about it in plant physiology is through this concept called water potential. And it's symbolized by psi, the Greek letter psi. And water potential is this idea of there's different forces that are acting on that body of water, whether it's the soil or inside the plant. And this water potential is essentially a difference in energy of the water. You know, when you look at 2 different spots in the soil, for example. And this water potential concept helps you figure out how water is going to move. In this case, in the soil, how's it going to move? And we say that water moves from a high water potential to a lower water potential. That's the mantra, from higher to lower. And an analogy they often give when you're first learning this is the idea of a waterfall. When you are watching a beautiful waterfall, water doesn't flow up the waterfall. It flows from a higher point of the waterfall to a lower point of the waterfall. And so you have a higher water potential at the top of the waterfall and lower water potential at the bottom. So water potential is a convenient way to think about water movement and how water will move. It is measured in units of pressure because that makes it very easy to actually measure physically. Like we have instruments that can do this, and it's very convenient and pretty accurate. And so pressure, usually we talk about megapascals is more commonly done, although some people still use the atmosphere pressure unit, but I tend to use the megapascal, or sometimes you drop the mega off and it's just Pascal, PA. So it just depends on the system and what you're measuring, if it's going to be mega or just regular Pascal. So it's pressure differences that we're thinking about. So from a higher water potential pressure to a lower water potential pressure is what we're thinking about. And if you want to know the conversion, by the way, 1 atmosphere is equal to 0.101 megapascal. All right, so in the soil, when we're looking at water in the soil and we're thinking about water potential, the water potential is going to be impacted by gravity. It's going to be impacted by what's called matrix potential. Matrix is thinking about the soil particles and how water interacts with the soil particles, as well as how the water interacts with other water molecules. So matrix potential is encompassing both adhesion and cohesion. And then we also have to think about this idea of, in that water, it's not pure water. There are minerals absorbed or dissolved, minerals dissolved in that water. So then we start thinking about an osmotic potential. Anytime you have solutes, whether it's minerals or sugars or whatever, anytime you have that dissolved in water, that creates an osmotic potential, right? And so you have to appreciate that the fertilizers that they apply to ag fields are salts. They're solutes. And so fertilization schedules and how they fertilize their fields profoundly impacts the osmotic potential. All right. And so, okay, so on Wednesday, we're going to get more into this. This topic is a little complicated. If you've never been introduced to this topic before, please spend some time studying this, because it takes a few runs at it before it starts to sink in. Okay, so we'll go into it in more detail on Wednesday.